Hey guys, I'm Christina and I'm going to be helping to talk through some of the processes of DNA extraction that we're doing today. So the first thing we're going to talk about are some of the materials that we may need to do this. So this little container of crushed ice is going to help keep all the things that need to stay cold in there. And this is a microcentrifuge where we're going to be using when we do to spin out some of the various solutions and pellets that's going to come up. And the range of amounts that we need to deal with in this experiment is anywhere from 2 to 1,000 microliters, and these 3 pipettes will take care of all of those sizes. And each of us is going to have a tube rack with tubes ready to be put in the microcentrifuge when necessary. We're going to have a permanent marker to label our tubes depending on amounts and specimens. And these plastic pestles here we're going to be using to grind up the tissue in the solution. And this is a heating block. We're going to be using that to store at various temperatures unnecessary. The last thing actually is, this isn't on the list. This is a waste bucket where you're going to put all your dirty tips and your solutions, supernatants that you won't need. So that's it. Hey guys. So I just finished dissecting a little sample from my little bug here. So this sample is going to be smaller than a grain of rice. I don't know if you can see, it's a little pink blob at the end of my forceps here, but it's going to be approximately 10 to 20 milligrams in length. So what we're going to do now is we're going to take this little bit of a specimen here and I'm going to put it in a tube. Now I've already put 300 microliters of the lysis solution in the tube, if I can get this sample. I'm going to take this pestle and I'm going to forcefully grind the sample up with the lysis solution. Now what the lysis solution is going to do is it's going to rupture the cell membrane and all the DNA genetic material is going to come forth, which is what we want. So I'm going to grind up this sample here with my pestle for about 2 to 5 minutes. And then I'm going to place it on this heating block here, and it's already at the 65 degrees Celsius, and we're going to keep it there for 10 minutes. Okay guys, so the next step is we are going to transfer our 2 samples that we have here from the heat block into the centrifuge. Now what's going to be accomplished through this is we're going to separate the liquid from the solid matter. The liquid is called the supernatant, and that's just going to have the genetic material and the excess lysis solution floating around in it. And the bottom is going to be a little clump of solid matter, and this is called a pellet. So what's in this is usually cell debris and other various forms of protein that might be in there. So we're going to take these from the heat block, and we're going to open the lid of the centrifuge here. And what's really important here is you make sure the rotor is fastened, otherwise bad things will happen. And we're going to place these 2 tubes with the hinges pointing outward like this, and they need to be balanced, otherwise they could fly out. And we're going to place the lid back on top, make sure that's fastened tightly. And with the settings here, our first little rotation will be centrifuging at 1 minute at maximum speed. So 13,400 RPM is going to be the highest setting for this particular machine, and 60 seconds here. And you can toggle up and down to get both of these options. So now we're going to close centrifuge and we're going to press start. Okay, so the centrifuge has stopped. So what I'm going to do is I'm going to remove my sample from the centrifuge. You can see we have a bit of a small pellet and we have the liquid stuff around it. So what we're going to do is we are going to transfer 150 microliters of the supernatant, which is the liquid solution, into a new test tube, which is right here. So I've set my micropipette here to 150 microliters, and I am going to transfer it now. So we're going to press it to the first stop, put it in the solution, making sure not to disturb the pellet at the bottom. We're going to let go, and that's 150 microliters of solution in the tip right here. And I'm going to press it all the way down to the first stop now, and all the way down to the second stop after to get rid of the very last drop here. And then what I'm going to do is I'm going to eject my tip into the waste bucket. So our next step is going to be, we are going to add 3 microliters. We have a new pipette for this one. 3 microliters of silica resin to our tube of the supernatant that we just collected. So what this is going to do is it's going to bind to the charge molecules. Pull up some, it's a very small amount this time. Transfer it right over to here. Stop. Okay, so I just switched to a larger volume pipette, so to make it easier to pipette up and down solution. So I've just finished pipetting up and down the samples to mix. As you can see, they're cloudy, both of them, and sometimes it looks like there are little particles floating around in them. So what I'm going to do now is I'm going to put them back on the heat block. This time it's at 57 degrees Celsius. So I'm going to put them on, and I'm going to keep them there for 5 minutes. Okay, so our next step will be putting the 2 test tubes that we have into the centrifuge. We're on step 9, as you can see. It's always a good idea to kind of check off steps as you go so you don't get lost, confused. So what we're going to do, take the 2 tubes off the hot plate. I'm going to open the centrifuge. It opens like that. Remove the top. Place them with the hinges pointed outward in the balance form. What we have here, we have it set. This time we're going for 30 seconds, so I toggled that down already and at maximum speed. So we are going to start. So now we are going to remove our 2 samples from the centrifuge here. I'm just going to place one over here and one over here. And the next thing with this one, there is a pellet at the very, very bottom. You can see that now. So we want to keep the pellet this time because this time the genetic material and stuff that was in the supernatant earlier, it's now in the pellet. So now we don't need the supernatant anymore. So I have my waste bucket right here, and I'm going to use the pipette to remove the supernatant without disrupting the pellet, and just very carefully. Alright, should be good. And then we're going to eject the tip into the waste bucket. Okay, so our next step would be to add 500 microliters of an ice-cold wash buffer. So I'm going to take our wash buffer from off the ice. So this will come as a concentrate, but your teacher will add ethanol to resuspend it. So what this does is it clears away all the contaminants from the pellet that we've already gotten. So all the nucleic acids will still be sticking to the resin, but this will just serve to clean it out, make sure it's good. So we are going to add 500 microliters. Set my micropipette. So. And now we are going to pipette up and down to break up the pellet. And if the pellet doesn't break up right away, you can use a smaller yellow-tipped pipette to do that, because it's just this stage is really important that the pellet breaks up. So, okay. So I placed my 2 samples into the centrifuge again. Hinges pointed outward. Make sure it's in the balanced configuration, and we're going to do this for 30 seconds at the maximum speed. And we're going to, yep. Top back over and start. And we're going to do this whole washing procedure with including both the wash buffer and the centrifuge twice. So you can refer to your instructions for the logistics on this, but you need to remove all supernatant from the solution that you have after you've done all the washing procedure. So we're on step 15 now, if you're following along with your instructions. So what we're going to do is we're going to add 100 microliters of distilled water to the silica resin. I just got to get my yellow tip out here. Sorry about this. Okay. we're going to add it to our pellet that we have here, and we're going to mix well up and pipette up and down like we did earlier. Like that. Good. Eject the tip. What we're going to do now is we're going to put this in the incubator here, and it's still set at 57 degrees, and we're going to keep it there for 5 minutes. Okay, so I'm taking these out of the centrifuge. We kind of didn't show the last step here, but we're on step 17 now. So we have a supernatant and a pellet once again. I'll get this one in here. So what we're going to do now is we're going to transfer 90 microliters of the supernatant to a brand new tube. And we are going to not disturb the pellet at all. We're just going to keep the supernatant in here. We can discard the pellet. And then after that, we'll store the tube on ice until we're ready to begin PCR. So let's get the supernatant. Check that. Tube, put it on ice, and store until ready to begin PCR.